The goal of this procedure is to show how polarization-based, total internal reflection fluorescence microscopy is implemented for the study of membrane remodeling during regulated exocytosis. This is accomplished by first ensuring optical elements, namely quarter-wave plate and polarizing cubes, are correctly positioned to generate discrete P and S excitation polarizations. The second step is to verify that the P and S beams are collinear, pass through the optical axis of the microscope, and illuminate the same portion of the viewing field when in total internal reflection fluorescence, or pTIRFM. Next, the cells to be imaged are stained with the carbocyanide dye, dye D, and stimulated with a depolarizing stimuli to trigger exocytosis. The final step is to capture images of fusing dense core vesicles while in pTIRFM. Ultimately, pTIRFM is used to acquire images in real time to monitor the membrane topological changes that result from chromaffin cell exocytosis.
The main advantage of this technique over existing methods like amperometry and membrane capacitance is that pTIRF reports a membrane curvature and, by extension, fusion pore dilation directly. The steepest part of the learning curve with this technique is first learning to align the P and S excitation polarizations so they are collinear, travel through the optical axis of the microscope, and illuminate the same part of the imaging field. A second issue that often arises concerns cell staining, namely how long to expose cells to dye D and what constitutes a well-stained cell versus one that is poorly stained. Demonstrating the procedure will be Tejeshwar, a graduate student from our laboratory.